Well, as part of our study of cytology, we now want to move away from changes in chromosome number to changes in chromosome structure, right? I talked about the fact that when we talked about cytogenetics, we were often looking at either the number of chromosomes or the structure of those chromosomes. And so we're going to look at this, and again, we're going to look at it from the standpoint of aberrancies, things that are abnormal types of forms that tell us about chromosomal structure. So, I mean, we physically already talked about how chromosomes are put together. We talked about nucleosomes and all the condensation that goes on to produce these metaphase chromosomes and beads on a string and that sort of thing. What we're talking about now is, okay, what happens to the DNA or the basic components of that? Now, with the change in number, and of course, euploidy or aneuploidy being in humans, you saw dramatic effects. Here you can see as severe dramatic effects from changes in the structure, depending on what genes or how much of the chromosome is involved, all the way down to certainly non-detectable. I guarantee you that we were looking at my DNA versus your DNA, we would detect some chromosomal structural differences. Okay, these are fairly minute differences, but they're there. They would be deletions or insertions, etc. And they have, without the ability to do it at the molecular level, that was simply non-detectable. But that can range all the way up to very severe and, in fact, lethal types of conditions. All right, we're going to look at three major types, and we're going to divide this into three lecture sets. The first ones, and the ones we'll be looking at in this lecture set, are called deletions. We'll then look at what are known as duplications, and then we'll end up by talking about what are known as rearrangements. Okay? Deletions are what they sound like. We're going to delete pieces of chromosomes, not entire chromosomes. So we're not talking about aneuploidy or euploidy. We're talking about a piece of a chromosome. A duplication is duplicating a piece of a chromosome. And the rearrangements, we're not going to lose any DNA per se. We're going to just move it somewhere, either on the same chromosome or between chromosomes. All right, so let's take a hand and look at deletions. We're going to divide these up into two primary types. One's we'll call a terminal deletion, which occurs on the end, losing the ends of a chromosome, and the other will be an internal deletion called an intercalary deletion, where we lose somehow some piece of the middle of the chromosome disappears. We're going to start by looking at terminal deletions, and that's simply illustrated here by the loss of an end of a chromosome. One end or the other of a chromosome will be lost, shortening the chromosome, removing some of the code, genetic code from that chromosome. This is just a little way we might illustrate it in a drawing. Here's our centromere. Here's some genes or some regions of the chromosome. Obviously, be lots of genes in part A as well as B, C, and we simply break off and lose part of it. Okay, and have a form. Now, something very important: chromosomal regions that don't have centromeres tend to be lost. So if this part fell off and it doesn't have a centromere, it will basically be degraded. Now we also have other issues with this, which is the fact that that chromosomes now have telomeres on the end, protective regions, and these don't have that, so they may be affected as well. But generally, the regions that are going to survive are those regions which have a centromere. A piece that doesn't have a centromere is generally lost. It's either lost by being attacked by the cellular system itself because it's not recognized as a chromosome, or most certainly by the time you go into mitosis or meiosis, there's no place for spindle fibers to form and connect, so you lose it in that position as well. Now these are sort of difficult to draw, so very often you see a problems kind of drawn like this, where we might see AB and some little thing representing a centromere, and then we say, okay, what's the difference? This is the condition we start with. What is this? Well, that would be a terminal deletion. Okay. Thought I'd give just a couple of examples of these. One we would call Creta syndrome, very one of the more notable types of deletion, at least a terminal type of deletion. And the second one is a very common one known as Fragile X syndrome, okay, which is a loss of part of the X chromosome. So within Creta syndrome, you get these individuals with very characteristic facial features. This is a deletion of the short arm of chromosome number 5 in humans. And apparently there are genes here mapped here that control the formation of the glottis and the larynx. Remember, those are our primitive areas here that were at one time gill slits, and now have become the structural units that produce the larynx, our voice box, as well as our glottis. And these are simply, this is a large chromosomal area, but we're only losing a small part up here at the top. and it is, as many of these will be, turns out to be in the form that we'll see it is a dominant, okay? Because you only have to be a heterozygote to express it, right? That's what a dominant means. So you'll get a normal copy, but you'll get this defective copy over here, and that's enough to cause these problems. That's called Creta because as infants, these individuals will cry, of course, but their larynx is not fully formed correctly, so they'll sound more like a cat, sort of when they try to cry. It's not a very good example, but you can hear these examples around. And again, this is just a terminal lesion, a small part affecting this arm, and that causes formation of this problem. Fragile X, much more of a common type of form. It's a loss of a terminal part of the X, and it can occur for multiple reasons. There can just be environmental reasons or whatever. But we found out fairly recently that there is actually an association with a large repeat region that occurs down here on the X chromosome. Okay? And so when that region breaks, that repeat region, you end up with a loss. And here's a nice little diagram of actually causing, and this would be a dyad, right? These are sister chromatids, and the loss is being caused here and captured by film. Now there are these CGG repeats that occur down on this part of the X chromosome in everybody. These are microsatellite repeats, so they tend to, because of unequal crossing over and from other things, but primarily unequal crossing over, they tend to grow or shrink, and they tend to grow or shrink by units of 3. So you might lose this part up here and become one that's just this part, or you may gain some extra. For people, Fragile X tends to occur more commonly when you have large elements of this. When these increase to great size and the repeats come very long, that becomes an area of the chromosome that will actually break. And we'll talk about other ways that Fragile X can occur later, but for us right now, and there's some methylation, all kinds of things can happen down here. But for us right now, as far as just a deletion, the longer these sequences are, the more likely you are to see a terminal deletion there, a Fragile X result. So it's familial. There are certain families that have very long stretches of these repeats and other families that are fairly short. And those families that are very long, you're more likely to see Fragile X appear in the pedigrees over time in different generations more commonly. Okay? Now, what do we get from all that? Well, we have this DNA repeat region. If we lose the X chromosome, you get a number of very characteristic things. You get a very characteristic facial structure. Okay. We get some impairment ranging from just learning disabilities and speech issues all the way up to fairly severe mental conditions where there's, very much like in Downs or something on the extreme cases of those. They have a number of things that are very characteristic as far as their behavior. They will often have attention deficit and they will be very hyperactive individuals. They have anxiety and unstable moods, so they swing, so they'll be sometimes happy and sometimes not. They have these autistic-like behaviors, these sort of repetitive type behaviors that you might see here, or this young lady that you might see here trying to do things, and relates, of course, to social interaction capabilities, etc. In fact, 20 or so years ago, when Fragile X was kind of being recognized, it thought that this might be one of the major causes of autism. It turns out that it's not. You get autistic-like behaviors, but this is not the cause of what we're seeing in this rise of these autisms that we're seeing nowadays. Okay? But as far as the morphology, they do have these long faces. They tend to have large ears. They have hyperextensive joints, so they can bend their fingers back in great degrees that other people can't. And they have all these sort of issues that can occur. Now, it's most common, of course, in males. It's fairly rare to see it in females. And you say, well, why? Well, just think about it. It's X chromosome, right? So the males only have one copy because they have a Y over here. So if you lose a large chunk of the X chromosome, then you're very likely that it's going to affect males more than females because females have this other X. And of course, they have the lion hypothesis and all of that. So we lose the X, one of the X's anyway, in females. So they potentially will displace for the most part one of these X's that has the problem. right? So those are two good examples of terminal deletions. There are many others. If you want to look through de Gomper's book, you can see that there's lots of these terminal type deletion effects that are going to occur. The other form we see are intercalary deletions. These are the internal ones, and in this case, you lose some part in the inside, but you keep the ends. Okay, and these actually are fairly common and that to persist because what happens here is that you're actually keeping very often both the centromere and both telomeres. So if the telomeres are retained, then it's a fairly well protected chromosome. Now the piece that's lost here is usually gone. I mean, we're gonna see that piece. If it doesn't contain a centromere, particularly, it will be lost. Now your question could be, well, how does the middle of a chromosome just fall apart, and then these pieces join back up? Well, there's a couple of different ways this can happen. One is we'll see later is unequal crossover, but another common way is simply remember that when DNA, particularly when it's in G1, it's just beads on a string. It's a fairly loose consistent. Well, it can loop and do things, right? Well, if you have a loop and you tend to have a break somewhere in that DNA, multiple things can happen. One is you can fuse it back together. There's repair mechanisms, and they'll fuse it back together correctly. So when you pull it apart, everything's in good shape. Okay. Another thing that can happen is they get put incorrectly. We'll see later that's called an inversion. And another thing can happen is that these ends, and when DNA breaks, it's called sticky. The ends of DNA are sticky. They like to push back together with other pieces, either through enzymatic activity or just on their own to reform bonds. Well, these sticky ends sometimes will fuse back together like this, and these ends will then be repaired, and you'll end up with a shortened chromosome and this thing called a ring chromosome that looks like this. And that's usually lost. Now, if the ring chromosome contains a centromere, you'll actually end up with this structural chromosome that can persist for a long period of time, much like this. Okay, so this is one of the ways we can get intercalary deletions. We'll see that there are other less complicated ways that we can also get it, particularly during crossover and recombination events. Now, one of the ways we detect these is when we do a karyotype, is that you'll see these structures called deletion loops, right? Because the basic rules are this, and when these chromatids go into meiosis and they go into prophase and they synapse, the synapsing has to occur in a very regular way, meaning that the centromeres will line up with each other, and then the chromosomal regions will try to do their best to line up with each other. And again, being anthropomorphic, but they'll try to line up, right? Well, if you've got a shortened one like this example here, everything's fine. A can line up, the B region, the centromere, C, D, etc. But when you get to E, well, that's not the right component. This is a G component down here. In order for this G to get with that one, what'll often happen is there'll be twisting and bowing, and this larger one will bow out, and the G will pull in, as you kind of illustrated here, and you'll get this external loop out here. And that external loop will tell you that you've got a deletion going on over here. And it's sort of a nice illustration from a book that looks kind of like this. And that's what you actually see. You actually see sort of this bump out on these metaphase chromosomes or late prophase chromosomes. See, okay. Now this also can be caused by an insertion, we'll talk about later, but deletion loops frequently look like this. This is the way they synapse up. Okay. Well, just like we did before with the terminal deletions, I thought I'd give you a couple of examples of intercalary deletions. One that's fairly common is known as Williams syndrome. Williams is a small deletion that occurs on chromosome number 7. It impacts a elastin gene that apparently is found here, and it results, therefore, because elastin is critical for things like compression, movement of muscles and things. And one of the big muscles that has to do that a lot is heart. So these individuals end up having some heart disease. In fact, some of these athletic things where individuals have had suddenly ruptured hearts from being athletes, it's found that some of those individuals have Williams syndrome or some related form to that. Now, it's found, I said, on chromosome 7. Okay, and what you're seeing here is this is a metaphase or a chromosome, right? So late prophase, metaphase chromosome. These dots that stain up here are going to be the areas that represent the centromeres. And then what's happened with this is that they've put in a special dye that went specifically to the elastin gene area. And so you can see in this individual and in this individual, they were heterozygous for this elastin gene. This one has the elastin gene right there, and this one over here, and of course these are perfect copies, so when they copy the absence of the gene, the locus. This chromosome, this homolog over here, this dyad pair, no longer has the elastin gene. There's been the deletion. So this individual, again, Williams, is a dominant in this form, an autosomal dominant. So by being a heterozygote and not having it over here, that dominates over having it over here, and they end up with having the issue. If you're interested, there's some very good websites on Williams syndrome. They talk a lot more about some of the other developmental issues and things that go on with these individuals, and social issues, etc., that you kind of might want to take a look at. All right. Now, one other one that's really interesting as well is this Kagami Agata syndrome. Hopefully, get close to pronouncing it correctly. And this has been recognized fairly recently as a true syndrome, but there are a number of effects. One of them is that at birth, and certainly for some time, you have this sort of bell-shaped thorax. It looks, the example is shown here very clearly. It's kind of like a coat hanger. It's called a coat hanger form. It looks something like this bell-shaped thorax, and it will eventually fill out somewhat as the organs develop. But you have that. You have a hairy forehead. You also have this protruding area right here, the philothrome. That's a good example here. This protrusion below the nose and ears and above the top of the lip. This protrudes. A good example right there. They have the microganglia as well, and then they have these abnormal wall effects, and the walls being the walls found in here in this form. So we have sort of all of that going on at one time. All right. What's interesting about it to me is that this is a maternally related problem, and it is microdeletions that are found on this chromosome in the mother, right? And they're microdeletions. They're small little deletions, and there can be multiple forms of them. And the multiple forms end up giving you different levels of the syndrome themselves. So the mother will have, and they tend to be completely de novo forms of these mutations, although they can be familial in the sense that the chance of them occurring can be over and over, but it's a maternal form. Now, where is it? Well, it's over here on chromosome number 14. Okay, and it's an intercalary deletion that occurs way down here on the end of chromosome 14 in this region down here. It turns out that the deletions impact somewhere between 24 to 26 genes are found here, and there can be various forms of them. And it turns out the gene region here for the females, this is paternal and this is maternal, are involved with things like the placental relationships as well as some of the neural systems. And so if the deletions are there, you end up with effects in the brain in particular. And it's not just one type of deletion. There actually are a number of different forms that can occur, microdeletions, and they've been mapped out in their effects both on the body as well as placental formation. You can see here deletion 1, deletion 2, as well as what are known as epimutations, which are new mutations that can occur. Okay, the deletions obviously are ones that will be more likely to be passed down, right? Even though they can occur spontaneously, the mutations themselves are almost all spontaneous random mutations. So you see a big mix there, and you see lots of different effects by these. So this is one of those areas where we know they have the mutations, we know they have the deletions, but there can be a wide range of outcomes from those. Okay? Another example, classic example of intercalary deletion is Prader-Willi syndrome. Okay, this Prader-Willi syndrome is a deletion found up much higher, but is on chromosome number 15. And these are caused by deletions that occur on the paternal chromosome. Remember, this is a case of genetic imprinting. I keep coming back to that over and over as we've learned more and more about it. But this is a case where the father is sending down signals for early development that occur right here in this area. The signals are located here. or the signals all over the chromosome, but this is one of the areas. If this is deleted, if there's an intercalary deletion, so you're missing the paternal information, then you end up with what's known as Prader-Willi syndrome. Has lots of effects. One of them is that there is excessive hunger, and these individuals are hungry all the time, have difficulty controlling their eating, so they've become often morbidly obese at an early age. They have very poor uh, structure of muscles and controls. They often have this staggering gait that this individual is showing you right here. And they have a poor suckling ability. They have all sorts of things that can occur, that issues just from having a deletion here. Now, so mother's providing fine, but it has to be the paternal side where you end up with this effect. And again, very definite facial features, lots of different things that occur from it's a fairly large deletion, but it occurs up in this region. What's interesting about it is if we go to the chromosome 15 on the maternal side, we get a completely opposite outcome. Okay, we get what's known as Angelman syndrome. So if you go on the maternal side, okay, and you have mutations or defects there, you have imprinting defects, you end up with what's known as Angelman. So now you've got the two homologs here. One of the imprinting that occurs on one, if it messes up, you go toward Prader-Willi. If it messes up on the maternal side, so both are active, you end up with what's known as Angelman syndrome, which are these children that look like they're happy all the time, etc. The reason you have that is that there's a gene activated on the mother's side that's very important in neural development. And it turns out that when this gene is, there's a defect with that gene, you get these problematic issues with in neural development systems, and it leads to all sorts of things. Now, one I skipped over. And is true for both of these as well as many others. It's one of the way we know these occur is through what's called uniparental disomy. I mentioned that very briefly when we were first talking about chromosomes. I said there's disomy, right? Well, uniparental is examples where uh, individuals get all the chromosomes they're supposed to, right? So you get 23 pairs, you get 46, except they're not the proper pairs. For some reason, you end up with two maternal, say 15s, or two maternal 7s or whatever, or two paternal 7s. That's called uniparental disomy. You're diploid, but you didn't get one from one parent and one from the other. There's multiple reasons this is thought. Probably these are cases where it should have been a triploid, but it threw one out. Well, it threw out the fathers and kept the mothers or whatever, and that can occur. And Prader-Willi, one of those uniparental disomy is one way that we can see Prader-Willi. There's no real deletion at all. It just turns out that the individuals get both of the 15 chromosomes from mom, right? So there's no dads there to place. So you end up as Prader-Willi. With Angelman, it's just the opposite. If you end up with two paternal chromosomes, there's no maternal one there to work. And so it's not the chromosomes work fine. It's just that you didn't get the proper number, and you can actually end up with Angelman. So that's not a deletion at all. Obviously, that is a different condition, but it's called uniparental disomy. It's one of the ways that you can get either Prader-Willi or Angelman syndrome, or in fact, many of these others can be driven the same way. Okay, well, those are all the examples that I really want to go through on deletions. So we have terminal and we have intercalary. We have some nice examples of where these have effects on humans. The next series will come along and we'll start talking about duplications, where we've added DNA to a chromosome itself.